us this afternoon. For those of you who I don't know, I'm Staci Kallish, and I am the president of the board of directors for NTSAD. And I am happy today to welcome all of you to this webinar. I'm really excited that so many of you are here today. You may know that NTSAD has supported research for many years, driving forward work on treatments for all of our diseases, including late onset Tay-Sachs and Sandhoff. This upcoming clinical trial from IntraBio investigating IB1001 is an exciting advancement. We're pleased to work with IntraBio on bringing this informal session to you and on supporting you and your families through this process. We hope to have more webinars in the months to come, so stay tuned for those. Today we have Taylor Fields from IntraBio and Dr. Tatiana Bremova from University Hospital in Bern, Switzerland as our panelists. The presentation from IntraBio will take about 20 to 30 minutes, and then will be followed by about 10 or 15 minutes for questions and answers. You may see at the bottom of your screen, if you are logged in by Zoom, under the More button, there is an option for chat, and you can send your questions to any of the panelists so that we can answer those during the question and answer period. Feel free to send them whenever you're ready. Just so that you know also, we are recording this session, recording the video portion for the webinar with the informational part, but we'll not be including the question and answer session. And then we'll be sharing the video on the NTSAD website within the next week or so. With that, I'd like to thank Taylor and Dr. Bremova for their time today as they speak to you about the IB1001 compound and the clinical trials that are coming. Taylor, I'll let you take it from here. Thanks.
Staci, and thanks Diana and Sue and everyone from the community. I've had the privilege to present a few times in various settings and at the NTSAD conference, but it's great to be here today. I'm the least important person on this panel. I'm going to let Tatiana introduce herself. Tatiana is a neurologist. She is a phenomenal advocate for rare diseases, and she also is one of the scientists who actually discovered N-acetylleucine and was the one responsible for identifying its benefits for GM2. So we're thrilled to have Tatiana here today and to talk through the science with us.
Thank you very much, Taylor. Thank you very much, Staci. I'm very happy being here with you today. Just a few words to my medical background. So I have been working since 2012 with patients with lysosomal storage diseases. That was also a topic of my PhD thesis. I have been focusing especially on Niemann-Pick type C disease at the beginning. And I will be also speaking how we got to acetylzoleucine in patients with GM2 gangliosidosis. And that was basically through patients with Niemann-Pick type C. Currently, I am since 2018 in Bern, and I'm watching also a new research group here focused on rare diseases, especially on lysosomal storage diseases. Thank you very much for inviting me.
So I'm going to just introduce IntraBio quickly, and then I'll let Tatiana just share some of her disclosures. But IntraBio is a very small company. We're based in the UK. Our founding scientists are pharmacologists from the Department of Pharmacology at the University of Oxford, and then neurologists from the University of Munich. We have a number of small molecule drugs that we are developing, but we are prioritizing what we very creatively call the IB1000 series, which is N-acetylleucine in its various iterations, and what we'll get into today. Tatiana has, as I mentioned, been involved with the development, but she is not affiliated with the company. And Tatiana, if you want to share your disclosures quickly, that would be great.
Well, yes. So I have been working as a consultant for Actelion and Sanofi Genzyme, but I have no financial relationships with the company IntraBio. I have nothing to disclose, basically.
So you have unbiased questions if you'd like to ask them at any point. So we're here today to talk about our lead compound series, N-acetylleucine. N-acetylleucine, as we mentioned, was initially a target for MPC. And it was via that lysosomal storage disease channel that we eventually discovered GM2 as well. But what I think is most important to explain about N-acetylleucine is it really is 3 compounds. You can have the L enantiomer, the D enantiomer, or you can have a mixture. And we love to use the metaphor of the hands because just like your left and right hand are not exactly identical, the compounds are not identical and do different things in the body. Similarly, when you have the racemic mixture, so the overlap of the left and right compound, that is a different formula and it does different things in the body. So when we talk about N-acetylleucine, we really are talking about 3 basic compounds, L, D, and DL. And I'm going to hand it over now to Tatiana to discuss the initial development, which was with the DL, and how we got to where we are.
So thank you, Taylor. I will be speaking about the initial development of how we got to acetyldoleucine in patients with GM2 disease. So basically, acetyldoleucine is an acylated derivative of the essential amino acid leucine. It has been used to treat acute vertiginous episodes since 1957 in France. Clinical experience has shown that it has a very safe, very good safety profile, and there are no adverse events. So it is very well tolerated. Possible side effects comprise gastrointestinal side effects such as diarrhea or obstipation, vertigo, and sometimes fatigue. So how we got to acetyldoleucine in GMT. So around 2012, we have been working with acetyldoleucine with tanganil in patients with peripheral vestibular syndrome with peripheral vertigo, and tanganil has been basically on market since 5 decades in this indication, and it has been produced by the company Pierre Fabre to that time point. So we were focusing on peripheral vertigo. We have tried this in a mouse model of peripheral vestibular deficit, and what we have seen was that part of cerebellum, the activity or the glucose metabolism of cerebellum improved after administration of acetyldoleucine. That was how we got to acetyldoleucine in cerebral ataxia, signs and symptoms. 2013, Michael Stroop et al. Published a paper, a study on cerebral ataxias of heterogeneous etiologies, where they showed the benefits of this treatment, especially in the domains such as stents, gait, cerebral tremor, risk of fall, dysarthria, fine motor function, and so on. And to this time point, I have been working with patients with NPC, with Niemann-Pick type C, which is otherwise zonal storage disorders, focus on the oculomotor function of these patients. So I've been traveling around with my video echographic equipment, and basically since the hallmark of this disease, around 70% of all patients from the International NPC Registry as due to 2013 feature cerebral ataxia, our intention was, like, the first one was, what if, even though NPC is very heterogeneous, acetyldoleucine really indeed works in NPC? And that was the case. So the 2015, we published on paper on that, also small case series in 12 patients with Niemann-Pick type C. And we saw not just improvement of cerebral ataxia signs and symptoms, we saw also an improvement of other NPC domains, such as dysphagia in 3 patients, social interaction has improved, caregiving, with other personnel in hospital. Also, like the performance of complex activities such as dressing, drawing, and like the daily performance has improved. So there was indication that there is the effect of acetylderuleucin also on other brain areas, not just on cerebellum, and that what led us to postulate that there is not just symptomatic, but there is also neuroprotective effect of the treatment. So that we do not just symptomatically treat cerebral ataxia signs and symptoms, but we also slow down the progression of the disease, we stabilize it, even though we do not cure it. However, the class A evidence, so a controlled clinical trial, being performed until now. So since we have been doing this for 7 years now, there have been more than 150 patients with LSDs around the world across a spectrum of lysosomal storage disorders and other neurodegenerative disorders who are actually on acetylderuleucin, and it establishes a good safety profile and also good efficacy. What I want to say is how we got to the GM2 patients was that we were just thinking, if it really works in MPC, what we were quite persuaded about, we postulated that it might be the case also in GM2 gangliosidosis patients, and that's why we decided to launch this clinical trial. So here in these videos, you see a mouse model of GM2 gangliosidosis, so-called Sandhoff mouse. On the left side, you see an untreated 13 weeks old mouse, and on the right side you see the treated one. And as you can see, the treated one, her motor function of her limbs and height and the tail as well, that is very important sign, is much better than that one not treated. So basically there is also visually, and also like the Sandhoff mouse that is treated lives longer than the untreated mouse. So these are basically benefits of the treatment in a mouse model that we can basically translate also onto the patient. So here you see a patient of mine, late onset patient of mine with late onset Tay-Sachs disease. On the upper video, you see baseline performing 9-hole peg test, and on baseline, and on the video below it, you see after 4 weeks of treatment. And basically, what you can see is that the performance has improved. She is quicker on that. So if you can imagine, like there are some complex activities during the day that you struggle to perform, that are difficult to perform because of just not cerebellum, but because also like different muscle tones, if it is a hypo or hypertonia, if there is like muscle weakness and so on. So what we have seen, there was an improvement of the performance of complex activities, and we have seen improvement of the scales, cerebral ataxia scales and scores. So if we have a look on the tab on the graph in the left corner, so the SARA, that is a scale for assessment of ataxia, that is 14-point scale, and the higher the score, the more impaired the patient is. And what we have seen that the SARA has improved, so the patient. That was also the case in performing of 8 meters walking test. This is a domain of spinocerebellar ataxia functional index, Scaffi, and at so patients were quicker while going. They were more stable, basically. So what we have done so far, we have administered acetyl-D-leucine to patients with late onset, but also late infantile patients.
If you want, I can jump in too, and you can go
Yes.
grab the charger. I'll do that if you like, because I can explain why we transitioned. Hopefully, when Tatiana's back, I can ask her to fill in. So Tatiana,
thank you very much.
So in parallel, IntraBio, the company, while all of this fantastic compassionate use data was being generated, while we were doing the in vivo, so the mouse studies, and in our labs doing the cellular work to further explore and support the proof of concept efficacy data that we saw in the clinic, we also were working to identify the optimal formulation. So as Tatiana said, Tanganil was approved in France in 1957 for acute vertigo. It is and will forever only be an acute vertigo treatment approved in France and 3 former colonies. To develop the drug, it would always need to go through a rigorous development program. And we didn't want to set out to develop a drug that wasn't optimal. So we've spent the past few years exploring the different effects of the 3 different molecules, as I said, what the best formulation is, what the best, the combination. And what we've identified is that N-acetyl-L-leucine, so the L enantiomer, is responsible for the effects of the racemate, the DL. But beyond that, it not only is responsible for the effects, we see that the D enantiomer has potential antagonistic effects when administered independently and on the racemate. And interestingly enough, the reason that we know this is we see the drug has many different biological effects, so different mechanisms. All 3 of these compounds have effects, positive effects in mice models for the short-term treatment of ataxia. But when we look at the long-term treatment, when we look at the disease modification, the survival, the functioning long-term, we see L works the best, we see DL works okay, and we see that D is antagonistic. So again, that tells us that there's different mechanisms responsible for the effects, but the long-term administration, the neuroprotective, the disease-modifying benefits are coming from L, and that D is inactive at best and antagonistic potentially. Because of that, we are prioritizing developing N-acetyl-L-leucine as an independent compound. We are formulating it as an oral solution, so it's eventually a sachet that will be swallowed, because we know a lot of patients with GM2, with NPC, with other lysosomal storage disorders have swallowing problems. An important thing is that there's no gluten in the mixture. It might not seem like an important thing, but we've seen with Tanganil that there are, especially for GM2, a lot of adverse reactions, not a lot, but some adverse reactions, and we think it's to the binder, the gluten binder, that are rashes and, or as Tatiana said, occasional vertigo or tremor. So we are formulating N-acetyl-L-leucine, again, creative, we call it IB1001, and that is the lead compound that we're now testing and which we're using in our clinical studies. So Tatiana, if you want to discuss, I can also discuss, but N-acetyl-L-leucine, as I said, is the active ingredient. It's an oral solution. It is not currently available anywhere in the world except in our development program, and we have full safety data on it. We know that it's equally well tolerated, has a very benign safety profile. It also has disease-modifying neuroprotective effects as well as symptomatic benefits.
Well, exactly. I would like to comment on this. This is important, Taylor, what you said. We need to emphasize that basically in this clinical trial, Lform, so the more pharmacologically active form will be administered. This form is not available currently on the market. There is no drug that could be administered, that could be taken. So the L-leucine, we think that basically the Lform will be also like more potent, that the effects will be more robust. So even though if we have seen effects with acetyl-D-leucine, but we do not know what happens with the Lform, we can just speculate. So I would say I would be rather optimistic, But just to make it clear that the DL form is not the same. So the drug, what is currently available, not in this indication, in indication of vertigo in France and in former French colonies, is not the same what will be administered in the trial, and probably is also not that potent, not that strong than the drug that is being developed. That is very important. Also, another thing, if some of you are already taking Tanganil, you know how big the tablets are. And this is also like acid. It's like amino acid. It has like this a bit. I have tried it, obviously, just to know how it tastes. So yeah, it's like the taste is not very good. I mean, it's acid, obviously. So that's nice about the thing that the formulation will be different. It will be liquid. There will be no choking. You do not have to like smash it together. You know what I mean. You do not have to like make it smaller. I don't know the word, Taylor, but how do you call that? To meals?
Grind it up.
Yes. Yeah, you do not have to do that. So that's really a huge advantage, I think. Yeah, and also you can just easily put it through pack, like gas tube, through tube. That was not that easy with the tablets, obviously, as well. So I think, and gluten, obviously, like to get rid of gluten is a huge advantage as well. Yeah.
So I think the important takeaway here is we will be the first, IntraBio is the first to admit that the development history of this drug has been a unique pathway. And it is unique in the sense that we are so fortunate that Tatiana and Michael Strupp were able to begin the initial investigations with the racemate, with DL. We never would have had this potential treatment for GM2 if it wasn't available. But it has always been and still is a development program. We need to demonstrate the drug works in a controlled clinical setting. And just like any other company, we also are going to develop the best drug for patients. So it is, I know it seems like a transition from something as Tanganil and or DL. For us, it's always been N-acetyleucine and figuring out the best version for it. And so I hope that it's clear that the drug that we're doing now is what we believe to be the optimal, most effective version for the treatment of GM2, that we think will have the most profound clinical benefit, that has been formulated for the patient population, and that we still very much need to demonstrate in a controlled clinical setting is effective. So I think we're going into the trial design now, which is good timing. So that brings us to the clinical study that we're running. We're running a multinational clinical trial for the treatment of GM2, so Tay-Sachs and Sandhoff. This study is run in the US, the UK, Spain, and Germany, and we're looking to recruit 30 patients. The study is the culmination of the last 7 years of work, and what we've seen in the compassionate use programs, our work with organizations like NTSAD, with clinicians, with the community, and we've tried to design the program so that it is able to demonstrate a clinical benefit, but also appropriate for the patient population. And that it sounds nice, but it's taken a bit of time to get there. And what we've eventually come up with is an innovative study design and novel primary endpoints. So there was a lot of back and forth with the regulators about that, but we're so thrilled that it has now been approved and active, and that the regulators were extremely willing to work with us, because they recognize that in order for treatments to get to these patient populations, we need to design studies and we need endpoints that are appropriate for your community. So the study we've designed, again, is based on all of that. We have 2 enrollment pathways, quote unquote naive enrollment pathway and a non-naive enrollment pathway. The reason that's there is that we are aware patients are taking whatever legal form of the drug. If it's the DL, if it's L, we know that there's off-label use of acetyleucine, and we did not want to limit enrollment because of that. So for patients who at their first visit to the clinic have been using any form of acetyleucine in the past 6 weeks, we ask that they complete a 6-week washout. And that's really important, because if we want to demonstrate the benefit of N-acetyl L-leucine of IB1001, we need to compare it to something. And if we compare it to a similarly pharmacological ingredient, that doesn't work. So we need to generate a true baseline, which is why we asked to do the washout. We've tried to keep the washouts as short as possible. At this point, a year ago, we were meeting with the FDA to discuss a double-blind placebo crossover study. We cut that because the feedback from the community was, if it was 6 weeks of treatment for 6.5 months of no treatment, patients were not interested in that. It wasn't feasible to recruit. And so we heeded that very strong feedback and designed a program that we hope is more appropriate. So we've cut the placebo. It is an open label study. All patients will receive treatment for 6 weeks, and then it's followed by a 6-week washout. And the reason that we can do the study like this is, one, we know that the drug has very quick symptomatic effects. So in as little as 2 weeks to 4 weeks, we've seen very significant benefit. And we didn't want to run a study that was longer than it needed to be in regards to the symptomatic benefit. We also know that the washout, that when patients stop taking the drug, they return to a form of baseline. So they see the benefit, and when they stop taking it, the symptomatic effects dissipate. And so that allows us to use patients as their own control, which is really important. And so although the study is run open label, we have the baseline period, we have the treatment period, and we have the washout period. And what we're doing is we're using novel primary endpoints, which we call the clinical impression of change in severity, to ensure that the primary outcome measure is blinded. So for patients, for their clinicians, you will know when you're on treatment, you will know when you're off treatment. What we do is we take videos at every visit. And we have gone a step further, and we take videos of 2 symptomatic scales, the 9-hole peg test, which you saw earlier, and the 8-meter walk test. And the reason we take videos of these 2 scales is because we know that no 2 patients are alike. Some patients may have issues with gait, with walking. Some patients might have issues with fine motor skills. I would love to see the scale expanded one day to include other symptoms, but the arrangement we had with the regulators was, because it's novel, they wanted us to start with really validated symptomatic endpoints. So the patient's first visit to the clinic, it's, instead of me sitting behind a computer screen selecting the 9-hole peg test as a meaningful scale for every patient, the patient, your doctor, will decide which test is more important. They'll select either the 8-meter walk test or the 9-hole peg test as an anchor test. And again, those are then videoed at each visit. They go into a very complicated algorithm, and 2 blinded reviewers will get the videos, and they'll ask to make comparisons. They won't know the time point, they won't know when the video was taken, but they'll look at the start of treatment, after treatment, and after washout. And they're looking at all aspects of functioning. So I'm just gonna go to some videos now which help explain this. But the videos and this design is important for a number of reasons. One, it allows for different symptoms to be assessed, things that are hopefully more clinically meaningful for each patient. It also allows us to have broader inclusion criteria. So patients can do either or. If patients are unable to walk, they can do the 9-hole peg test. If they're unable to do the 9-hole peg test, they can do the 8-meter walk test. And that allows us to broaden the inclusion criteria. And that's important because based on our clinical experience with this drug, we've seen that there can be really profound change in short time, but we could exclude patients. So Tatiana, I'll let you explain this video if you'd like, because this is from Tatiana's published.
I probably will hear myself also in background exactly. This was a patient with late infantile onset Niemann-Pick type C disease. And she also suffered frontal lobe involvement, meaning that she had just like pathological crying or laughing, like very emotionally leaky. And prior to the treatment, she just stopped and started crying, like not able to do anything. And then you see On treatment, she was just totally changed. And obviously, there are fluctuations in the clinical state. I mean, you have to encounter that. But if you look at the video after 1 month of washout, so now on medication, she finished it in quite very time, because she wasn't that strike. She didn't have such striking cerebral involvement. Yeah, like cerebellum was not her problem. Her problem, like coordination was perfect, but her problem was frontal lobe and cognitive deficit. And after 1 month of washout, again, complete arrest. So you can see like the drug can work on different levels. This is pretty interesting, I think. And what is nice about this clinical trial is that we actually, we were involved in the designing of this trial, and in the designing of how to get, like what kind of anchor test and so on. And this is nice, this translational approach. What we have seen is like, it's so heterogeneous that at the end we wouldn't be able to really recruit the amount of patients, because like almost anybody would be really would really like be okay, this is the patients for the trial. No, so this is very nice, that term, that we like really try to also include patients, if it is possible, in quite late stages of the disease with different involvement. Yeah.
So one of the nice things about the endpoints is, if in a traditional study the primary outcome assessment was the 9-hole peg test, this patient wouldn't be eligible because she's unable to complete the test. In our study, the patient would be eligible because she's able to complete the walk test, but we can still capture a clinically meaningful change. And so that was something that was really important to us, is that although we might believe we know what the right question to ask is, because patients have such different symptoms, it's inappropriate of us to say it's going to be one thing for everyone. And we wanted to be able to demonstrate that there is variability, and that we need to tailor and we need to cater the outcome assessments on a more personal level to assess a clinically meaningful change. So similarly, that one accounts for the heterogeneity, but two, a really important thing we came to realize was that speed does not equate to a change in functioning. And that has been a shortcoming in a lot of the endpoints that have been used, that a change in speed, so doing a task quicker, doesn't necessarily equate to a change in quality of life. And so again, I'll let Tatiana talk through this video. It's one of her patients. This is a Sandhoff patient. And I'll just make a comment at the end. So Tatiana.
Yes, so this is a, exactly, late infantile patient with Sandhoff disease. Very cheerful, very, very, very nice, very cheerful boy. And we administered, he's still on acetyldolaleucine, and after symptomatic treatment, so after 4 weeks of treatment, he was just much more stable running. I mean, even though, he has also like musculoskeletal problems, contractures. These are always issues. Like if you have, like, this is the thing about these rare diseases, about these lysosomal storage diseases, for example. They are so heterogeneous. There are so many symptoms, and that's why if you want to, you cannot sometimes really validly evaluate one symptom without encountering another one. But still, you can see the improvement even though you have like also dystonia, spasticity, and. So yeah, he, like, the stability has improved, the risk of falls has reduced, and he still profits. He still benefits from the treatment. He's still on since 2017. Yeah, even though, the disease, we do not cure the disease sensu stricto. We slow down progression. We really, but earlier the disease is diagnosed, the diagnosis is established, and the earlier the treatment starts, the better. because you need the neurons that are still there, like, to be affected, you know. And at point when there are no neurons to work onto, no treatment would help. So the earlier, the better, always.
Yeah. And the final thing I want to say about this is, again, the change in speed, there isn't. We've seen the times for this. When he's walking with the assistance of his mother and when he's walking independently, there isn't a difference in time. But you can see in the middle video, he can't walk independently. The stance captures that. The change in quality of life and functioning from being able to walk unaided to independently is dramatic. And we wanted to make sure that we were capturing, again, all aspects of that in the study. And it wasn't just, is the patient walking faster or slower? So the trial itself, as I said, it's being run in the US, UK, Germany, and Spain. In the US, we have 3 trial sites. We have a site at NYU Langone, we have a site at Mayo Clinic and UCLA. NYU and Mayo are open and recruiting. UCLA, we think, will be open and recruiting within the next week. In the US, the eligibility criteria is patients 18 and older. I should add that the treatment is not just a late onset treatment. In Europe, patients 6 and older will be enrolled. That was a regulatory decision. It was the decisions made by national agencies, but we very much intend to develop the drug for the entirety of the age spectrum for GM2. Again, we're looking to recruit approximately 30 patients, and we're involving very standard scales. So ataxia rating scales, quality of life, we do video recordings. There are no invasive biomarkers. There's very limited blood sampling and urine tests. We know quite a lot about the drug safety profile, so we don't need to do anything too cumbersome there. And the drug is administered still in the dose for 13 and older of 4 grams a day, but it's a little bottle that you drink now instead of large pills, as Tatiana said. And so these are the clinical sites. And I just want to take a moment and comment on why the clinical trial is so important. So again, we have had the real benefit to have the opportunity to generate all quite extensive compassionate use data. We're running the trials for identical trials for MPC and a subtype of cerebellar ataxia as well. So there's 3 studies. We benefit there as well from the opportunity to have the compassionate use data, but the trial is an investigation. If we would ever like to have this be a treatment, an approved legal treatment that ensures its longevity, we have to have meaningful clinical data and we have to get the drug approved. And so the clinical development program is intrinsic to the success of this drug. The drug will not be here if we are not successful in our development program. And our development program is very much aimed to ask rigorous questions. Does the efficacy that we believe we've seen in all the proof of concept, is it there? And to do that, we have been so appreciative and grateful to work with the patient communities. We have been so lucky to have partners like NTSAD, and we really are always trying to make ourselves available so that you do have the information to know why we're making decisions, why things are going, and to help facilitate the development of the drug. So Tatiana, do you want to add to that at all?
Well, what I would like to add is maybe I will provide all the papers, all the publications, everything about the treatment to your community. Like Diana said, that she will put it onto the website so you can have a look on that. I really hope that the clinical trial will really generate. We will see the effect we think we see in patients in small case series, and yeah, we hope that we help indeed patients with GM2 gangliosidosis. Thank you.
So that's the end of our presentation. Thank you so much. I put my contact details up there. I know Tatiana's happy to answer any questions, but I will put anyone in contact then if you'd like to reach out to me. For patients who are interested in the study, the best thing I'm going to go back one slide. The best thing I can recommend is going to clinicaltrials.gov. If you can look up the full study procedures, you can look up the assessments, and then reaching out to the trial sites. So as industry, I have absolutely no influence, no decision making, wouldn't ever be involved in inclusion, exclusion. That's all the PIs at the trial sites. So if you do want to reach out to me about the studies, I will most likely put you in direct contact with the emails you see there. But you are welcome to contact the sites. They are, again, actively screening and recruiting, and more than happy to reach out. We have a travel policy which provides for whichever form of transport, an overnight stay if it's necessary, for meals, and that's for patient and one caregiver. And so we really are trying to help facilitate people participating in that. So I have a question already that says, anything in the works for early onset infantile Tay-Sachs? As I mentioned, we are very much hoping to develop the drug for all age groups. We have a lot of reports from around the world of patients as young even as 6 months who are taking illegal forms of the drug. And it is our goal to have it approved for all age groups because, as Tatiana said, we really believe the earlier intervention, the way that the drug works, if the neurons are still in very good shape, it can help keep them in good shape. Once a neuron dies, we can't bring it back to life, but we can help it go longer, for a non-scientific term there. But I'm going to now pass it on to either Staci or to Diana to facilitate the questions, and we'll try and answer as many as possible.